All right, today we're diving deep into one of the most amazing, fundamental processes of life itself, DNA replication. We're going to get right into the nuts and bolts of the molecular machinery inside prokaryotes, and we're going to use the classic lab workhorse E. coli as our guide. We're going to see just how a cell pulls off a seemingly impossible task, flawlessly copying its entire genetic blueprint.
So take a look at this. This is it. You're looking at the entire genetic instruction manual for an E. coli bacterium. It's just one single circular chromosome, but it's packed with 4.6 million base pairs of DNA. I mean, think about that for a second. If you were to type all of that out, you'd have a book thousands of pages long. And every single one of those letters is absolutely crucial for this little organism to survive. And that right there brings us to the central challenge, the big question. How on earth does this tiny cell duplicate that entire 4.6 million letter code in just 42 minutes? And not just fast, but with near-perfect accuracy. How does it pull off a feat of biological engineering that incredible? Well, that's the mystery we're here to unravel. So here's our game plan. We've broken this down into 5 parts. We'll start with the replication challenge itself. Then we'll walk through how the whole process kicks off. We'll see the machinery in action, understand how it's all controlled, and then finally, we'll compare this bacterial system to what happens in more complex cells like, ours.
Okay, first up, Section 1. The replication challenge. Let's really set the stage for this whole process.
The whole operation, really, it can be broken down into 3 main stages. You've got initiation, which is all about finding the exact starting point. Then comes elongation. This is the main event, where all the new DNA is actually copied. And finally, termination, bringing everything to a clean stop. All 3 are critical, but today we're really going to focus our attention on those first 2.
So let's dive into that first, and you could argue most critical step, Section 2, starting the copy. This is all about initiation, because let's be real, if you don't start in the right place, everything that comes after is just meaningless.
You see, replication doesn't just start randomly. Nope. It begins at a very specific DNA sequence, kind of like a unique street address. It's called the origin of replication, or OriC. In E. coli, it's a 245 base pair region. And if you look closely, you can see it's not just a random string of letters. It has these very distinct sections, these specific binding sites. Think of them like reserved parking spots for the proteins that are going to kick this whole process off.
And this slide, I love this. It lays out the whole initiation team perfectly. It's a whole crew of specialized proteins, and each one has a very precise job. You've got DNA A, we're calling it the key master because it recognizes and opens the origin. Then there's DNA B, the helicase, which is basically the unzipper that unwinds the DNA. It gets put on the DNA by its escort, the DNA C protein. Primase acts as the primer painter, laying down a little starting strip for the process. SSB proteins are the stabilizers. They're crucial for keeping the unwound strands from just snapping back together. And finally, you have DNA gyrase, the swivel, which is so important for relieving all the tension that builds up as you unwind that helix.
So here's how that whole team works together. It's like an ignition sequence. Step 1. The key master proteins, DNA, they bind to their spots at OriC. And when they do, they actually cause the DNA to bend, and that strain physically forces a small section to pop open. Step 2. As soon as it's open, the DNSC escort jumps in and loads one DNA B unzipper onto each of the separated strands. Step 3. And they're off. These 2 helicases just speed away from each other in opposite directions, creating 2 replication forks that are going to travel around the entire chromosome. Okay, so the DNA is open, the forks are moving, and that brings us to Section 3, the replication fork in action. Now we get to see what's happening inside the molecular copying factory itself.
And this is the factory, a massive molecular machine called the replisome. It's not just one enzyme, as you can see, it's a whole complex of proteins all working in concert. You can spot the helicase up front, unwinding the DNA. And right behind it is the main copying enzyme, DNA polymerase. See that ring-shaped protein holding it on? That's called a beta clamp. It works like a carabiner, just locking the polymerase onto the DNA strand so it's super efficient and doesn't fall off. This whole machine just parks itself at the fork and synthesizes both new DNA strands at the same time.
Now here's a really crucial point, but it can be a little tricky. The 2 strands of the DNA double helix are anti-parallel. So think of it like a 2-lane highway where the traffic is flowing in opposite directions. One strand runs in what scientists call the 5' to 3' direction, and the other runs 3' to 5'. Here's the problem. The DNA copying enzyme can only drive in one direction. It can only add new letters to the 3' end. So this forces the cell to get creative and use two totally different strategies to copy both strands at once.
And this diagram just visualizes that challenge perfectly. Look at the top new strand, that's the leading strand. It's being made smoothly, continuously, as the fork moves along. Easy. But now look at the bottom one, the lagging strand. To copy this strand, the DNA has to be looped out so the polymerase can actually build backwards, away from the fork, making these little chunks. Each one of those chunks needs a brand new primer to get started. And later on, another enzyme called DNA ligase has to come in and stitch all those little pieces together into one continuous strand.
And these short DNA segments on the lagging strand, they actually have a name. They're called Okazaki fragments, named after the scientists who discovered them. They are the fundamental, disconnected little building blocks of that lagging strand. The replosome just churns them out one after another, and then DNA ligase comes along behind it to seal up all the gaps.
So let's circle back to that speed we mentioned at the very beginning, because the numbers here are just staggering. You've got a 4.6 million base pair genome. It gets copied in 42 minutes, that's 2,520 seconds, and remember, there are 2 replication forks moving away from the origin, so each one is only responsible for copying half the genome. So how fast is that machine actually moving? How many letters is it adding every single second? The answer is roughly 912 base pairs per second, and that's at each fork. The replosome is literally racing along the DNA, reading the template, and adding almost 1,000 new letters to the growing chain every single second. The speed and the precision needed to do that are they're on another level. It's truly mind-boggling.
Now, when you have a process this fast and this powerful, you absolutely need tight control. Which brings us to Section 4. Controlling replication. How does the cell make sure it only copies its DNA once, and only once per cell division?
Well, the answer lies in this really elegant biological timer. It's based on a chemical tag system called DNA methylation. Essentially, the cell marks its own DNA with these little methyl groups. And the status of those marks, right at the OriC origin, determines whether a new round of replication is even allowed to start.
Here's how that methylation clock actually works. Step 1. A fully methylated origin. That's the go signal. Replication can start. But as soon as that fork passes through, you get to Step 2. The DNA becomes hemimethylated. That means the old strand has the marks, but the new one doesn't. This state triggers Step 3, which is like a safety lock. A protein called CQA comes in and binds specifically to this hemimethylated state and physically blocks a new round of initiation. It's only after a delay, in Step 4, that another enzyme, methylase, comes in, methylates the new strand, and basically resets the origin, making it ready for the next cycle.
So that is the beautifully efficient system in E. coli. But how does this stack up against more complex organisms? That brings us to our final section, A Tale of Two Cells, where we'll compare that prokaryotic simplicity with eukaryotic complexity.
So while the basic principles are the same, you still unwind the DNA and copy it, the actual machinery is different. You can see here, instead of that DNA protein, eukaryotes use a big multi-protein group called the origin recognition complex, or ORC. They use different DNA polymerases like delta and alpha instead of polymerase 3. They have a totally different system for removing the primers. And because their chromosomes are linear, not circular, they have a unique problem, copying the very ends, which involves a special process and structures called telomeres.
And this image, this right here, shows the biggest difference in strategy. A bacterium like E. coli has one origin for its 4.6 million base pairs. A human cell, on the other hand, has 3 times billion base pairs. Trying to replicate that from a single starting point would take weeks. So eukaryotes solve this by having thousands of origins of replication. Replication just kicks off at all of these points at once, creating thousands of these replication bubbles, just like the ones you see here. They expand and merge together until the entire massive genome is copied in just a few hours.
The key takeaway here is just the sheer scale of it all. An average eukaryotic cell doesn't just have a little more DNA, it has 25 times more. This massive leap in the amount of genetic information required a complete overhaul of the replication strategy, from using thousands of origins to employing much more complex and specialized protein machinery.
And that really just leaves us with a final thought to chew on. We've just walked through this incredibly sophisticated, high-speed, high-fidelity copying machine in what we consider a simple bacterium. It really makes you wonder,
If this is the simple version, what kind of even more incredible and complex machinery is at work inside every single one of our cells, flawlessly copying the 3 billion letters of our own DNA right at this very moment? It's a process fundamental to our very existence.